W teacher.
All right, so I have handed back exams. I will take questions in just a minute here. Overall, you did very well. You did better than my class last year did. Better average. Grades were spread across, which is pretty typical for a first exam. This is combining data from your class and the class before yours. So overall, you all did very well. And if you didn't do well, that's okay. This is only one exam, only 45 points. Now you know my exam structure, how to study a little bit better, maybe what to do differently next time. And I am happy to talk about exams at any point. I will say I'm human. I make mistakes. I had to grade 72 of these. All right, there's a lot of room for error there. Check to make sure I added everything up correctly, right, and that I didn't miss anything. If you did that About Me questionnaire, you got some points back for like visiting office hours or tutors. If you sent me a sloth, that was that Easter egg in the syllabus, you also got that point back too. If you think you sent me that and I did not mark it on your paper, let me know as well. All right, in terms of questions, so I like to do stats, right? I will calculate how many of you got it wrong, right? So the percentage of the class that got a particular question wrong. And my philosophy is that if it's over 50% wrong, that means either I did not teach it well, the question might have been worded wrong, something was going on, all right? I like to give you a chance to get those points back. So 2 of those questions, fill in the blank 5 and multiple choice number 9, were 2 that were very commonly gotten wrong. Now because this is the first exam, I also included anything that 40% of you got wrong, which would include short answer number 8, that table that you filled out, fill in the blank number 2, and multiple choice number 11 there. Right? Those are very commonly gone wrong, right? I understand why some of them were wrong. I think I might have worded something off on one of them. On your diagram with the organelles, I put storage of lipids, and a lot of you answered smooth ER, which is true for lipids, but the main function there is storage, which is not the ER. So I want to give you points back opportunity. So what I do for this is that I have you hang on to your exams. Don't erase anything that's incorrect, but rather for these 5 questions, correct it on your exam. On another sheet of paper, write me a sentence or 2 as to why that, what your rationale was for choosing the correct answer. And then you'll turn in the hard copy back to me. Right? So I'm not going to do a Moodle submission because I want to get your exam back once you correct it. If you don't need to correct anything, don't want to correct anything, that's fine. The exams are yours to keep. You don't have to turn them back in. Even if you correct something and turn it back into me, I will hand it back to you eventually here. This is just some way that you're not losing out on points for whatever exam. Any questions? Still have a question or did I answer it? All right, so the hard copies of your exams with your corrected answers will be due back on Friday, and then I will update your grade up on Moodle. All right, there's no more exam questions. Like I said, I'd be happy to meet with anybody who wants to talk.
What's going on with pancreatic cells?
Pancreatic cells?
Yeah.
You're going to have to look that up because that's part of the points back that you get. Yeah. Alrighty, I will say if you are already at 100% and maybe that, like the sloth point got you there and you were correct another one, I'm not going to give you over 100%. Alright? But that means you don't have to worry about it. You can still correct it so you know. Alright? But. Alright, shifting into Unit 2 content. On Friday, we talked about glycolysis, which is the first step in cellular respiration. Right? But there's actually 4 stages. So we're going to go through the last 3 of those today. Right? before we even dive in, Friday was 2 whole days ago. You've had a weekend in between. Let's do a review. So I want you to answer the questions. What are those stages of glycolysis? What does each stage produce? And what is the net overall ATP and NADH yield of glycolysis?
Oh, what's that? Would be 2 NADHs and the 2 NADHs. Oh, yeah, 2 and 2, yeah.
Or breaks down the carbon to make.
I see the wrong version. So it produces what it is.
Alright, before we go on to the review, I know I had a question about the video lecture from Friday. It is on the YouTube channel, but the link on Moodle was not correct, so I have to update that link. Any of you ran into that problem? I've had a couple of you tell me about it, which thank you for telling me about it, because otherwise I would have never known to correct it. Alright, group review. What are those 3 stages of glycolysis? Energy investment, cleavage, energy liberation. Yes. And what is produced during energy investment?
Fructose 1.
2 ATP is used, but we're going from glucose to fructose 1,6-biphosphate, right? During our cleavage stage, what is happening? Yeah, we're breaking apart that fructose into what molecule? Glyceraldehyde 3-phosphate. And our last stage, energy liberation, what is the end molecule produced? Pyruvate. And what is produced along the way in terms of energy molecules?
2 NADH and 2 ATP.
We have 2 NADHs. We're producing 4 ATP, but overall the net yield is only 2 because we used 2 of those up in that first stage. All right. I've not given you this slide because I literally just copied and pasted it from your Friday lecture, just as a review in case there are any questions here. But again, big picture here, we're ending with pyruvate. We are obtaining 2 ATP, 2 NADHs. All right, again, this is a slide from Friday, so I'm going to skip forward.
Wait, so you said the net yield is just the end, basically?
Yeah, so the net yield, so like the gross yield would be 4, but we're using 2 up, which means the net yield is only 2 ATP. So we're only considering those that we have in excess. All right, before we move on from glycolysis, I just wanted to mention a couple of things. These are not things that I'm going to test you on, all right? But they're rather good examples of what's happening. So I know we talked about feedback inhibition when the product inhibits itself from being made. We see that in glycolysis. We see that in step 3. So remember, there's 10 steps broken into 3 stages. Step 3 is part of the first phase, energy investment phase. And there's an enzyme in this step, right? Phosphofructokinase, right? It's a mouthful that catalyzes the reaction here, right? It's this enzyme that will be inhibited during feedback inhibition. So we're producing a lot of ATP in glycolysis. Once those ATP levels become pretty high, and we don't no longer need to go through glycolysis, ATP will inhibit that enzyme in step 3. So this is a really nice example of feedback inhibition. And then another fun fact that I thought was worth sharing with you all, especially if you're looking towards going into the medical field at all, is that cancer cells, they tend to have high levels of glycolysis. For whatever reason, they tend to go through glycolysis really rapidly and not so much the rest of cellular respiration. And we can see this when we do certain types of scans on patients, right? So this is a type of scan that was done where we can see high levels of the enzymes used in glycolysis, and they happen to be accumulated around where the tumors are, indicating that these cancer cells are highly utilizing glycolysis over other types of other cells around it or going through the rest of cellular respiration. It's called the Warburg effect, right? Again, I'm not going to test you on this, but I thought it was really interesting.
I was just gonna say, should we write this down in this letter?
This is just kind of for your information, trying to relate it to, especially those going in the medical field, something that you could come across. All right, so glycolysis. We have created 2 molecules of pyruvate. Those 2 molecules are going to move on in cellular respiration into the next step. And the next step in this case is the breakdown of pyruvate, right? Now notice we're going from outside of the mitochondria to now the rest of the steps are happening inside of the mitochondria. All right, we're going to break this down to more specifics here. I do like this image because it's a really simplified way to look at cellular respiration and where it happens. Well, let's look at the breakdown of pyruvate. All right, so this is step 2. Pyruvate has been produced, right? We know that the rest of this reaction is going to happen inside of the mitochondria, so we have to get the pyruvate inside of the mitochondria. And I like this image. It does a good job of showing you how this happens. So there's an outer membrane channel. Remember, mitochondria have a double membrane system because of the endosymbiotic theory. So that outer membrane channel, and then to get in the inner membrane, it has a symporter, which means we're transporting 2 molecules across at once. One of those molecules is pyruvate. The other is a hydrogen ion, or a proton is another word for that. All right, so pyruvate, we have gotten it into our mitochondria, right? We are now going to break it down. So pyruvate is going to be broken down via a specific enzyme, which is pyruvate dehydrogenase, right? Catalyzes this reaction. And what it does is it helps remove a molecule of CO2, right? So in this image, our pyruvate, half of our molecule is in blue. This blue part is not going to change, right? But the CO2 that's on top of this blue highlighted area is going to leave, right? So we're producing CO2 here. We're also going to add in another molecule that we call coenzyme A. We're adding that molecule to what's left of our pyruvate, right? So pyruvate comes in, a molecule of CO2 is taken off. What's left is an acetyl group, right? That's what's highlighted in blue here. That acetyl group will combine with another molecule, right, called coenzyme A. And what we get is acetyl-CoA as the final molecule produced in this stage. Right, so in the meantime, we produce molecules of CO2. We also produced some NADH. Right? NADH is also produced during this step. So in terms of energy-containing molecules, this step yields one NADH for each pyruvate. Now remember, one glucose molecule yields two pyruvates. So per glucose molecule, this would happen twice, once for each of those pyruvates. So per glucose molecule, this would be 2 NADH. All right, let's go over this again using a slightly different graphic, different wording. All right, so again, we have pyruvate entering into the mitochondria. This image is only showing you one transport protein when there's actually two there. Once it enters in, a molecule of CO2 breaks off of that pyruvate. What is left is an acetyl group. Right? Pyruvate dehydrogenase is the enzyme working here. Right? It helps to make NADH plus a proton. And it adds in this coenzyme A molecule to the remaining bit of the pyruvate, to the remaining acetyl group, and we get acetyl-CoA. You know, I'm throwing a lot of terminology at you on a Monday morning, so bear with me. All right. Let's step back and review. This is a table that we're going to keep filling out throughout the rest of the lecture. Great study tool. Right? So I want you to, for glycolysis and the breakdown of pyruvate, tell me where does this occur? What is the end product of this stage? And what is the net yield of energy per one glucose molecule? Feel free to work together. We're going to keep filling it out. I would write it down, or I don't know if you like download the slides. That saves a lot of handwriting. Write that down. Type on the slides.
Yeah, that's stage
Kind of personal preference.
and the breakdown of pyruvates stage 2. End product of the glycolysis would be 2 NADH and then 4 ATP but 2 ATP. And then and then the net yield would be 2 because there's 2 NADHs in glycolysis.
Your family begin to do your family trauma? Yeah.
That makes sense.
It's so like messed up though.
Yeah, I have that same feeling. Like, yeah, something's off. Yes, what's created that would go on to the next stage? Glycolysis, what is the molecule created? But it's not the end molecule. It's transform glucose into something else. Yep, yep.
Are we going to do this for all 4?
Yes, yes.
Nico,
All right, let's finish our review together here. Where does glycolysis occur? In the cytosol, yes. My clicker is not working today, so I have to stay by my computer. All right. What is the end product of this stage? 2-pyruvate. Yeah, pyruvate is produced. That'll go on to the next stage. And what is the net yield of energy-containing molecules per one glucose molecule? Yeah, exactly. 2 ATP and 2 NADHs. Now, our next stage, breakdown of pyruvate. Where does this happen? In the mitochondria. Yep. What is the end product of this stage? Acetyl-CoA. Yep. And what is the net yield of energy-containing molecules? Yeah, 2 NADH, 1 for each pyruvate produced, and we know we have 2 pyruvates per 1 glucose molecule. Yeah. Exactly. So once glucose splits into 2 pyruvate, there's essentially 2 lines of reactions happening at the same time. All right, let's move on to stage 3. Citric acid cycle. So acetyl-CoA has been produced. The next step is for that acetyl-CoA to enter into the citric acid cycle. Right? Now, unlike the other 2 stages, where we start with something and we end with something, right? This is a cycle. So we're going to start with a molecule, and we will end with that same molecule so that the cycle can continue. All right, so big picture here. Citric acid cycle. It's a cycle. Some molecules are going to enter, others are going to leave, right? We are going to produce CO2 as a byproduct, as well as some ATP, as well as some NADH. And we're also going to produce this molecule called FADH2, right? It acts in the same manner as NADH, right? It is an energy-containing molecule. It transfers electrons and protons. All right, let's dive in a little deeper even, right? There's a lot of steps that go on here. I'm going to tell you exactly what I would expect you to know in terms of the citric acid cycle. 2 of those molecules that I would expect you to know are called oxalacetate and citrate. So let's look at how they work in the citric acid cycle. We have our acetyl-CoA, right? That was produced at the end of the breakdown of pyruvate. It's going to enter into this cycle. When it does, it's going to lose the CoA, the coenzyme A part of its molecule, and it's going to combine with this molecule called oxalacetate, right? Once the acetyl and the oxalacetate combine together, they form the molecule known as citrate. Right? And this is where the cycle gets its name, citric acid cycle, because of this molecule, citrate. All right, so oxalacetate combines with the acetyl-CoA, we create citrate. That citrate is now going to be transformed into a series of other molecules in the rest of these steps here. I'm not going to have you memorize the rest of those molecules. I want you to know oxalacetate and citrate. Once it's citrate, it goes through the cycle. Right? As the cycle continues, we are producing, right, some CO2. CO2 is like a byproduct, right? We're producing CO2. We're producing 1 ATP, 3 NADHs, and 1 FADH2, right? Per 1 pyruvate molecule, or 1 turn of this cycle. Now, we have our glucose molecule, produce 2 pyruvates. So if I were to ask you how many molecules of the products are produced per glucose molecule, what would you tell me? Yeah, it's going to be doubled, right? Because this cycle happens twice per 1 glucose molecule, once per pyruvate. and there's 2 pyruvates per glucose. All right, so we would double that if we're talking per glucose rather than 1 round of this cycle. All right, so we're producing a lot of energy-related compounds here. Right, per 1 turn of the cycle, we're only producing 1 ATP. That's not a lot. But the big picture here is that it is a cycle, and this oxaloacetate will be regenerated, so it can be used time and time again. Right? So this last molecule that it ends up as is malate. Right? Malate can then become oxaloacetate, which can then start the cycle over. It can combine with acetyl, become citrate, go through the cycle, become malate, right? Become oxaloacetate, combine with acetyl, become citrate, right? And it just keeps continuing on and on, right? So oxaloacetate can be regenerated. That's a key thing when we're talking about the citric acid cycle. I don't want to go too fast because I know I'm throwing a lot of terms and numbers at you here. All right, so again, just reiterating here, right, that we have our glucose molecule, breaks down into 2 pyruvate, which yields an acetyl-CoA each, which means per glucose molecule, we have these things happening twice in steps 2 and 3. Right? So per glucose molecule, like we said, we would double it because there would be 2 acetyls going through per glucose molecule. We would get 4 CO2, 2 ATP, 6 NADHs, and 2 FADH2s. Just the citric acid cycle. I'm emphasizing this because this is something that I see a lot of students stumble over, is asking the question between 1 round of the citric acid cycle or 1 glucose molecule, right? You get different results. All right, so steps 1 through 3. We have produced some ATP, not a ton, but we produced some, right? We have produced carbon dioxide, which is really just a byproduct. We're not going to use it for anything. In fact, we will end up exhaling it out. But we produced a fair amount of NADH and FADH2s, right? Our energy-carrying molecules. Carries those protons and electrons. And through lab this week, I know CO2 is a byproduct, but it's a byproduct that you will measure in lab when you look at cellular respiration in cockroaches. Right? So we can utilize the byproduct to see, how much cellular respiration is occurring for many organisms. All right, we're going to come back to our table. Right? You can either rewrite in the first 2 columns, but I want you to focus on that last column, our citric acid cycle. Right? And then if you get through that, there's a lot of diseases, genetic diseases, that are known as mitochondrial diseases, right? It's mitochondrial function is impacted, right? So find a mitochondrial disease. What causes this disease? How might you see it in a patient? I'll give you a few minutes to go through this as well. And remember that last question is asking per glucose molecule.
Yeah, citrate.
And then
also have 10 NADH.
And the so
I can write on that.
Yes, you can write on that. Yes.
there's one.
after 2 seconds.
Yeah.
When they have high cholesterol, they can
Well,
That's a good question. I remember diabetes.
how many candidate molecules do
I'm not an expert on the topic.
Yeah.
So, there's that caveat, but that would be fine.
so
So,
is
thank you. Yeah.
so we have one
Over here, you don't have numbers.
so we have two NADHs here. Right, and then that takes it to here. And then just each pyruvate has a two NADHs.
You just say F828.
Yeah.
I don't know that last one.
Yeah, I'm trying to get understanding. Still trying to understand.
Yes. So net is like what the gain is. So if we think of glycolysis, we have 4 ATP. So that'd be like the gross yield, but the net yield would be what's in excess. Usually 2 of those ATP and we're producing 2 that aren't being used.
So the net yield would be what? Or what you
What you have in excess.
Oh, access.
Yeah. Yep.
Wait, so
The net yield is mindset glycolysis. For all the other steps, you're not using what you're producing.
okay, so
Two,
these
so
were all
then
still.
4 CO2,
Yeah, yep.
Okay, because that's something.
2
So
ATP,
for this
6 NADH,
how much you have excess.
2
Yeah, how much are you producing and not using during that stage?
Okay, okay. That's not even-
that makes sense, doesn't it?
I think so.
All right, let's focus on this last column here since we've already done the first 2. Citric acid cycle, where is this happening in the cell? Also in the mitochondria, yeah. All right, what is the end product of this stage? It's kind of a tricky one. I would consider it oxalacetate, right? So unlike the other stages, we don't have a molecule that's being produced and moving on to the next stage. Instead, we have many energy-containing molecules being produced. But at the end of the citric acid cycle, we have oxalacetate that has been regenerated and ready to go again, right? What about the energy-containing molecules per 1 molecule of glucose? How many ATP? 2, yes. 6 NADHs and 2 FADHs. I also put 4 CO2 in there, so that really shouldn't be in there because this question's only asking about the energy-containing molecules. And CO2 is not an energy-containing molecule. But we are producing CO2 as our byproduct there. Did anybody have time to look up a mitochondrial disease that you would like to share about? Well, I'm not going to make you share. Yeah.
Leber hereditary optic neuropathy.
Okay.
What's that?
What is it?
It's a rare genetic disease that quickly and unexpectedly makes you lose your vision.
Oh, it's related to the mitochondria.
Yes.
Interesting. Yeah, so there's a whole host of diseases. You probably came up, if you got to this question, with like at least 20 different answers here. Again, this is not something I'm going to test you on, but rather something that kind of applies to real world scenarios here. All right, we're going to move on to the last stage. And in my opinion, this stage is the hardest to understand and learn, so we're going to walk through it rather slowly. All right. This is our last stage of cellular respiration. And what is going into this stage is not a molecule, right? So we're not having pyruvate enter the stage, we're not having acetyl-CoA enter this stage, but rather what's entering this stage is all of those energy-containing compounds that we have built up thus far. That would be our NADHs and our FADH2s, right? They are entering into this final stage, right? I'll elaborate on the next slide a bit. But this is also the stage that requires oxygen, right? So I mentioned glycolysis, it can happen without oxygen, right? This stage cannot happen without oxygen. It needs oxygen. And we will see why in the next few slides. So, NADHs, FADH2s are entering in to this stage. Right? Remember, they're containing protons and electrons that they can easily give away and accept. If we look at where this is occurring, right, it's in the mitochondria, but specifically it's happening across the mitochondrial membrane, right? So that inner membrane. Remember, we have two membranes, the outer and the inner. This inner membrane here is where all of this is occurring. This inner membrane has all of these different protein complexes that are embedded within the phospholipid bilayer, right? They each have their own name to them. I'm not worried that you know the names. Rather, I want you to focus on function here. So they're embedded in the mitochondrial membrane. And what happens is that our NADHs and our FADH2s, once they come in contact with these protein complexes, they are going to give away their electrons and give away their protons. Right? Because that's what they're meant to do. That's what these molecules do. They give and accept electrons and protons. In this case, they're giving them away. We can see NADH has turned into NAD+, plus two of these protons. Right? It's also given away its electrons, which in this image is denoted as these yellow stars. Right? And if we just pay attention to these yellow stars, right, we can see that they are moving through these proteins. Right? They are passing through all of these complexes embedded in the membrane. They don't just pass through, but rather they provide energy as they pass through these proteins for these protons to move across the membrane. Right? So we have these electrons, they're moving across. As they move, they're shuffling these protons, these hydrogen ions, across the membrane. And what we get is this buildup of a gradient. Right? We get a lot more of our protons on one side of our membrane than on the other side. Right? Now this happens in a series of redox reactions that, again, I'm not going to have you learn because I want you to focus on the concept here, not all the nitty-gritty details. Right, so again, we have these electrons. They've moved through these series of proteins complexes. As they move, they're shuffling these protons and building up a proton gradient, right? An electrochemical gradient of hydrogen ions. All of these H pluses, right? Now we know these H pluses, right, want to move with their gradient. Molecules want to move with their concentration gradient. And that is exactly what they will do in the final step of cellular respiration here, right? So it's this gradient that we have built up that's going to provide the energy for our final step. All right, before we talk about that final step, we're gonna take a step back, right? Because there's one more thing we need to mention before we move on. I told you this stage requires oxygen. Well, where is oxygen being used here? Right? Oxygen is being used as the final electron acceptor. So that electrons that have moved through these protein complexes, they have to end up somewhere, right? They're not going to stay free floating. They have to be captured by something, and they're actually captured by oxygen. And once oxygen captures those electrons, it's also going to take in a couple of hydrogen ions to make water, right? So we're producing water in this stage by combining oxygen with that electron that moves through our electron transport chain and a couple of hydrogen ions. So we would say oxygen is our final electron acceptor. Now, there are some organisms that use this to their advantage, one of which is the kangaroo rat, right? They live in the desert. They barely drink any water at all in their lifetime. They get all the water they need through metabolism, through this specific step, right? They don't have to drink because the water they need to live comes from, right, cellular respiration, which is pretty unique that we have animals that can adapt to using it in this way. Now, we need much more water than we can get through metabolism, which is why we need to drink way more. But organisms like this kangaroo rat, right, have this very specific ability. All right, we have our final electron acceptors, oxygen. We have created water, right? We have moved protons across a membrane to build up a gradient. The last step is that those protons want to move back onto the other side of the membrane to balance out the gradient. They're moving from an area of high to low, but the only place that they can do this is at this very specific enzyme, very specific protein that we call ATP synthase. This is the only way that our hydrogen ions can move back across. Now, as it moves across, it's actually powering this enzyme. And it's powering this enzyme to make ATP. So it's actually causing the phosphorylation of ADP. So remember, we can take ATP and turn it into ADP by breaking apart one of those phosphate groups. Right? We know those phosphate groups, those bonds hold a lot of energy. So we can break them apart. But at some point, we have to put it back together so we can get ATP, so we can use that energy again. Right? And this is the process that does it, right? Phosphorylation, we're adding in a phospho group, right, to a molecule. We're adding in another phosphate group. And it does this by using this gradient. As these hydrogen ions flow back across, flow into this protein essentially spins, right, and adds that phosphate group to ADP to create ATP. Right, so phosphorylation is the process of adding in a phosphate group to a molecule. Doesn't matter what molecule, as long as it's a molecule. But the process of actually making ATP has a different name. It is phosphorylation, but we would call it chemiosmosis. Right, that is the actual process that is occurring here. It's the production of ATP using hydrogen ions flowing through the ATP synthase. We could spend a week talking just about ATP synthase and how all of the subunits, you can see there's all these different subunits that are color-coded here, work to make this happen, right? But that's not what Bio-10 is about, right? Big picture here is that this is kind of the wheel that makes it all happen. As it spins, it adds that phosphate group. We're producing ATP, and we're producing large amounts of ATP, not just 1 or 2 molecules, but we can produce upwards to 36 molecules of ATP in this process. I put this next slide in here just to reiterate that this is a protein that spins. We actually know a fair amount about this protein. I'm not going to test you on this, but I think it's noteworthy to notice that it spins, and it's this process of spinning that attaches that phosphate group to the ADP. All right, I think this concept is hard to learn and harder to visualize. So I have a Professor Dave Explains couple minutes here showing you the movement of everything happening. Rarely, one molecule of glucose will be super efficient and channel all of that energy into making ATP, right? It's going to vary, and it varies for a variety of reasons, right? Sometimes the NADH produced is not going to continue on through cellular respiration, but go to another pathway, right? So thereby, that decreases the amount of ATP we will ultimately end up with. And sometimes that proton gradient can be used for other purposes within the cell as well. So again, less power for that ATP synthase, less ATP being produced. Which is why often you'll see a range here, right? Like 30 to 34, 24 to 28, 30 to 36 glucose, right? I tend to say upwards of 36 molecules of glucose because 36 is the largest range that I've seen. All right. I want us to fill out that final bit of our table here, and then we will review together. I did not put a timer on this slide, but let's do a couple of minutes here so we have time to go over it together and then discuss homework.
So this happens within Yep.
I guess I didn't see it. But now I was at
Okay. Oh, coming?
No, I was heard about it.
Oh.
It
But
is-
homecoming is
it's-
next weekend,
it is a
right?
chemical.
Homecoming is next weekend?
Homecoming is this week.
This weekend? All right. Well. Same science, right? Molecules and what they do. All right, I think it's been a couple of minutes here, so we'll go over the answers together. First of all, you have the first part of that table already filled in. Oxidative phosphorylation, our last step. Where does this occur? Yeah, in the mitochondria, but more specifically across the inner mitochondrial membrane, right? What is the end product of this stage? I mean, the end product is the end of cellular respiration. But this stage specifically, what is being made? It's kind of tricky. You could say ATP, right? You could also say water. Water is being made. But we're also taking away electrons and protons from NADH and FADH too. So we're also creating NAD plus and FAD plus, right? main product being made is actually ATP, right? And that is our next question here is how many molecules of our energy-containing molecule is being made? And what did we put? Yeah, 30 to 34, 32 to 36. It's a range, right? And I know I'm giving you different numbers, but as long as you give me a range that is within those numbers, right, I'm fine with that. All right. I wanted to go through maybe the last couple of slides here. Very quickly, right? Big picture here is that this last stage, we get the big ATP payoff. Oh, my pointer decided to work there. All right, we get a big payoff, right? 30 to 36 ATP. Most of our energy comes in this last step. Now, I wanted to note that other molecules besides glucose can go through this process. They're just going to enter into these stages at different points. So, for example, proteins might enter the stage at pyruvate. They might also enter the stage at the citric acid cycle. Right? So there are variations here. There's a lot to know. So I'm going to have you do homework, due Friday. I want you to draw this out and include these terms. Right? It's a great way to get some muscle memory in, to try to remember everything happening, put it on a piece of paper. You can either hand in a paper copy to me on Friday or upload it as a picture or PDF. I think I even allowed like JPEGs for this submission site up on Moodle as well. Beyond that, Wednesday we are going to do content check number 3. So have that completed minus part 2, and we'll do part 2 in class.
Hey, let's live. Come on.
And we still got out 2 minutes early.
For 2 minutes.